Tonight we have Alice Yuan Zhang, who joins us, a media artist who joins us from Los Angeles. And here physically in the room with me at Stanford, we have Jen Dionne from Stanford University and Ade Mabugunje, also from Stanford University. Each speaker will give a 20-minute presentation followed by Q&A. And so I will introduce each one separately. My introductions are notoriously very brief. Jennifer has done this 3 or 4 times, so she will not be shocked by my very brief introductions, bios introductions. But if you go to lasertalks.com, you find a link to their web pages and extensive bios. Jennifer Dionne is a professor of materials science and radiology at Stanford and a Chan Zuckerberg Biohub investigator who calls herself a sculpture of light at the nanoscale. She's also co-founder of the startup Pumpkin Seed, and I am honored of having known her for what, more than 10 years, I guess. And she's a brilliant scientist and a brilliant speaker, and she has spoken several times at the lasers. So we're curious to see what you've been up to since last time.
Cool. Thank you so much, Pierre. I'll unmute here.
Now what I have to do is kill my.
Cool. Actually, I might just bring my laptop up to get a slightly better view. That way those online are not seeing me look too scary. Maybe okay, cool. Everyone can hear me now and see me? Well, thanks all for coming out. It's exciting to be here in person. I remember when Piero organized these pre-COVID, and they were always amazing events, not only the smaller kind of salon style events on campus, but some of the larger events that you organize in San Francisco and San Jose. So it's great to see the community coming together. The first speaker was also, in some ways, somewhat inspiring. Like, I love seeing her art that kind of tries to merge both like natural and digital environmental factors and kind of express how that impacts humanity. Hopefully my talk is perhaps providing a more hopeful light on what technology can enable for personal and planetary health. And before I get into my presentation, I want to make sure I acknowledge all of the students and collaborators who made this work possible. Like Piero mentioned, I'm a faculty member in material science at Stanford. I also have a courtesy appointment in radiology, and I've benefited from many colleagues across campus in both Stanford's new School of Sustainability, as well as in the School of Medicine. And I thought I'd start with kind of a pop quiz. I know it's after dinner, but if you take a snowflake size amount of biological material, say just DNA, and you pack DNA into about 1 milligram or a snowflake size, how much data do you think is encoded in that small amount of DNA? Do you have any guesses?
10 petabytes.
Oh, that's a great guess. Thanks. Any other guesses?
1 terabyte.
Cool. Petabytes, terabytes. You're both not too far off, but still it's an enormous amount of data. It turns out it's an exabyte, so that's 10 to the 18 bytes. So when I say you're not too far off compared to most folks who kind of guess, you know, gigabytes, you're at least closer, but still orders of magnitude off. This is an incredible amount of data stored in such a tiny bit of matter. It's more data than that generated by Amazon, Google, Facebook, and Meta combined each day. And that data contains a lot of information with which to understand kind of the health of humans, the health of animals, the health of the planet. And yet it's really hard to read that data and kind of ascertain when there are transitions from, you know, wellness to a state of disease. And if you look at some of the best technologies we have out there to read DNA, like Illumina sequencers, they can read your gene sequences at a rate of about 400 bytes per second. So you compare that to the 10 to the 18 bytes that are in just a small amount of biological material.
How do you count one byte in this case?
Basically one letter of the ATGC code. And then if you compare that to average internet speeds, or actually 1 bit is 1 letter, so a byte is 8 letters. If you compare that to average fiber optic internet speeds, like for example in your home or using Stanford's Wi-Fi guest network, those are typically about 100 megabytes per second. So the rates with which we get information at the internet, orders of magnitude faster than our ability to read biological materials. And then of course, in biology, it's not just the DNA that matter. The DNA basically gets transcribed to RNA, and the RNA gets translated into proteins. And kind of our genes are fixed with time, but our proteins are telling us what's happening, with time and how our health is changing. And the fastest rate that we can kind of read or sequence proteins and how their concentration and form is changing with time is less than 1/10 of a byte per second. And of course, there are millions of cells and metabolites to identify as well. So what this talk will focus on is using some of the same technologies that we use for ultra-fast fiber optic internet communications, and using that to read biological samples more quickly. And the idea is basically to create a chip, much like the chip that many of you have in your pockets or in your cell phones right now, that essentially contains a bunch of miniaturized lenses that take light and then focus it down to molecular scale. So that way we can read what's happening in a variety of biological samples, basically on an output. And in that case, the output would just be a simple camera readout that tells us about the presence and identity of molecules that are near that silicon chip. And you'll see some pictures of the chip that we've designed. Essentially, you can think about it as a glass substrate, as a thin layer of silicon on top. And then Piero mentioned that we're kind of a sculptor of light at the nanoscale. We sculpt light by making essentially tiny little Lego blocks that precisely define what light is doing near the molecules. And in the talk today, kind of those Lego blocks look almost like these wishbone type structures. So when we illuminate the chip from behind, each of these serves like a magnifying glass that focuses the light to where the molecule is. And I don't go into too many details about how the structure works, but here are just some of the dimensions. For the most part, the dimensions of the structure are less than 10 microns in the long direction and less than a few hundred nanometers across. So quite miniaturized, about the size of a virus in this direction, and about 1/10 the width of a hair in the long direction. And part of the reason why we need to have these nanostructures on our silicon chip to do fast reads of biology and to focus the light is because many of the biomarkers we want to detect, whether those are gene fragments or proteins or metabolites, are much smaller than the wavelength of light. And it's kind of part of the challenge of doing very high fidelity molecular detection, and why most of our systems for reading biology are so slow. We can't see these objects, with our naked eye or even with the best microscopes, because the size of these objects is so much smaller compared to, most wavelengths of light that we'd want to use, say visible wavelengths of light. Of course, you can go all the way to X-rays to interrogate these molecules, and we have many collaborators who use the facilities up at, say, SLAC, the linear accelerator, which has kind of X-ray wavelengths to probe these molecules. But unfortunately, those wavelengths are so high energy that they actually wind up being destructive. So basically, we want to be able to see these molecules using visible light. So I mentioned that I wouldn't really go into the physics of how these structures work, but based on how we design the silicon blocks on our glass substrate, when we illuminate, essentially with a light emitting diode, the chip from behind, we can strongly focus light. And here we represent light by basically these E fields. So that is the electric field of light as experienced by the resonator or the silicon blocks that are on our chip. And I guess what's important to notice are two things. First of all, the spatial distribution of light. So you can see how light comes in and it basically gets focused to the central cavity region in both of these antennas. We have one antenna design where light gets focused roughly to about the size of a cell, and that is a structure we're going to use for detection of bacteria in wastewater, as well as detection of drug susceptibility of tuberculosis. And then we have another design where light basically gets shrunk down to almost this molecular scale volume. So here we have another notch or slot, and you can see this bright region in the middle. So the region with which light is getting confined is now comparable to the size of a molecule, like a protein. And in addition to the spatial extent of the electric field of light, you can also see that light gets enhanced quite substantially compared to the incident light that's on it. So you can almost think about this like if you've ever seen a solar concentrator, or if you've kind of focused light through a magnifying lens, like sunlight through a magnifying lens, you know that that spot basically can be very bright. So here what we've done is taken the incident light, and each of these lenses concentrates light to at least 1,000-fold compared to the incident light source. So they're very good at enhancing that field of light. And then this is a calculation. Don't worry too much about the particular trend, but if you look at the vertical axis, you can see the mode volume, or kind of the spatial extent of light compared to the wavelength of light here, lambda. So you can see that with the slotted structure, basically light gets confined to a size that's about 1/10 or 1/20th the size of light. So that's kind of like if we had a great shrinking machine that could take each of you and shrink you to 1/20th the size. So that way you could see, it's almost like a Honey, I Shrunk the Kids, right? Like if you remember that movie, exploring kind of new worlds once your size is shrunk down. So we have many sensors that we can put on a given chip. So here's a pattern of our chips. It's basically a glass substrate with silicon on top. And you can see that each of these sensors, when patterned on the chip, now has this very iridescent color associated with it. So they're actually really beautiful at refracting light. And we can pattern many chips onto a small area, or many sensors onto a small area. A 1 centimeter square chip can hold about 3 million sensors. And for anyone who's ever done biology, you know, a lot of times people spend time pipetting into these like well plates to run different reactions. Each of these sensors essentially corresponds to, or each 1 centimeter square chip corresponds to over 31,096 well plates. So if you wanted to do this sort of biology using conventional methods, you basically need the size of a basketball court to run as many different assays or reactions. So it's a highly miniaturized platform. And then in addition, what allows for kind of deployment in the environment, for example, for wastewater detection, or as I'll share with you, doing some detection of harmful algae blooms, is we can get rid of a spectrometer. So we can use a light emitting diode and basically sweep the wavelength of that LED. And here you can see a movie playing basically of the sensors lighting up as we're sweeping that wavelength. And basically each sensor here is tuned to a different wavelength so we can detect different biomolecules on it. So you can see the sensors are lighting up. You can also see the background is pretty much black. So even though we're on glass that's transparent, we've been able to design the optical setup in such a way where we're only seeing light that's coming from the sensor plus the molecule that's near it. And then I mentioned that you can think about these sensors as allowing us to do many reactions on there. What we need to do is be able to put different molecules in different regions of the chip to be able to sense many different biomarkers. And we do that essentially with a miniaturized printer. So if any of you have ever done inkjet printing, you know that each letter comes out and each letter is basically a series of tiny little dots. Basically, we've adapted that sort of technique, but we've made the dots now even smaller. So here, each dot that comes out of our inkjet printer, which is printing basically different molecules on the surface that allow us to target different biomarkers in the environment, is about 10 microns. So approximately the size of each sensor that's on our chip. And here's the Stanford logo. If you were to basically compare this, this whole image now is about the size of one dot that you would see if you zoomed into an inkjet printer in one letter. And then each dot basically puts down a particular surface chemistry over each resonator. So here we just have some fluorescent tags that show how the printer is allowing us to address individual sensors on our silicon chip. So let's put these silicon chips to work and show how they can allow for like very fast kind of readout of biological samples. And I know we only have about 8 minutes left, so I won't go into too many of the details, but first of all, I'll show you how we can do nucleic acid detection and also metabolite detection. And those are labeled techniques, meaning that basically on the chip, we've put down surface chemistry that's specific to each biomarker we want to detect. And we use this now for detecting harmful algae blooms. So I'll also share with you some of the underwater autonomous vehicles that we're deploying our chips on in collaboration with Monterey Bay Aquarium. And then I'll also share with you how we can avoid putting down any surface functionalization and basically use the chip to determine the drug susceptibility or the species and strain and drug susceptibility of bacteria. So in the nucleic acid case, what we do is basically put down single-stranded DNA that's complementary to the nucleic acid we want to detect. And many of you probably are now familiar with PCR tests. PCR is basically detecting nucleic acids. It does that usually by a chemical reaction that causes the target DNA you want to detect to be multiplied many times over and over with different changes in like the temperature of the reaction. In this case, we don't have to do that thermal cycling. We can just kind of directly detect the target nucleic acid, for example, like various SARS-CoV-2 genes, by looking at wavelength shifts in our resonators. And basically within a few minutes of adding the target, in this case, a nasopharyngeal swab, to the chip. So we add the target in here. We're detecting many resonators simultaneously, looking at different gene fragments from the SARS-CoV-2 gene fragment. And you can see within about 5 minutes of adding the target, we're able to get indication that a patient, for example, is COVID positive. And we're also able to do this with a very low limit of detection. So some of you might have used the at-home SARS-CoV-2 tests. Sometimes they have false positive rates if you don't have, or false negative rates if you don't have a high enough viral load. These sensors basically increase the sensitivity or allow for improved sensitivity by several orders of magnitude. So generally the sensitivity you get in at-home tests is about nanomolar. Here we have femtomolar concentrations, which is actually close to what you would get with PCR. So imagine an at-home test that's almost as good as PCR. We're actually super excited about this, not so much for SARS-CoV-2 and at-home detection, but for kind of correlating environmental DNA with various metabolites. And I should have put in an image here of kind of the Earth fluorescing from satellite images. Like if you look at images of the Earth at about 600 nanometers, you'll see fluorescence from the ocean that's coming from algae or phytoplankton. And phytoplankton comprise only about 1% of all plant biomass, and yet are responsible for more than half the Earth's oxygen production and a lot of important nutrient cycling. And of course, with climate change, sometimes the nutrient cycling can lead to overproduction of various small molecules like this toxin. So harmful algae blooms we know are occurring more and more frequently. And what we want to do is basically deploy these sensors to more readily detect harmful algae blooms and kind of correlate that with the environmental DNA that's shed by organisms in the ocean. So for the last year or so, I've been collaborating with NOAA and also Monterey Bay Aquarium to take our chips and put them into these underwater autonomous vehicles that Monterey Bay Aquarium has made. They have essentially an environmental sample processor. So all these little cartridges basically take ocean water, filter it, and then put it onto our chips, basically in this handling container that allows for kind of simultaneous detection of many genes from various organisms in the water, as well as detection of some of these metabolites for harmful algae blooms. So the hope is to have kind of an early warning or early detection system for harmful algae blooms and better understand both the physical and chemical drivers of those harmful algae blooms. And then just in the last couple of minutes, I want to share with you how we're using these chips now to monitor bacterial pathogens in wastewater. And many of you know that wastewater worked quite well actually during the pandemic to monitor viral levels. Basically there we're monitoring nucleic acids. It's actually much harder to do the same sort of monitoring with bacteria, just because there are so many species and strains of bacteria. And usually in environmental samples, it's very hard to do PCR. So the idea is we put the bacteria onto our chip. We illuminate it with a monochromatic light source like an LED or a laser. And then here what we do is basically detect the spectra that comes from that bacteria. So basically most of the light that gets scattered back, let's say we illuminate with a green laser, most of the light would be in the green, but some of the light is actually shifted to longer wavelengths based upon the molecular vibrations in the cell. Basically, the cell has its own vibrations at room temperature that change the energy of the incident light source. So what we've done is collect a variety of spectra. You can almost think about like the color, of the bacteria from over 31 bacterial species and strains. And you can see that the color of the spectra looks somewhat different between different bacterial species, but kind of similar between other ones. Like for example, E. coli and pneumonia look kind of similar in terms of their scattering spectrum. By eye, it's really difficult to differentiate, but we've been able to use neural networks from conventional kind of image processing algorithms, like in a Google image search, and be able to adapt them for these bacterial spectra. So what goes in is basically a cell with a spectra, and we have trained neural nets. So now when a new spectra comes in, we basically can say with some probability or some predictive score to what extent the new spectra is, for example, E. coli that might be in the wastewater or Pseudomonas pneumonia that might be in the wastewater. So here are some of the data. Basically, you want to have most of the numbers along the diagonal because that means when we know the ground truth that our neural network is predicting it correctly. So the ground truth usually comes from mass spectrometry, which is not a technique that can be easily deployed in the field. And each tool costs about $500,000. So what we did is trained the neural net on over 60,000 single cell spectra and tested it on over 12,000 spectra. And you can see we have pretty high classification accuracy. Here's kind of the zoomed out view of all 31 species and strains. Here, each color corresponds to the same kind of treatment that a clinician would use, like the same sort of antibiotic that a patient would be prescribed based on empiric evidence. So you can see most misclassification actually occurs within the same antibiotic treatment group. So we collaborated with Stanford's clinical microbiology laboratory that handles basically all patient samples and showed that when we trained our neural net, not only on kind of lab-grown cell lines, but also then took it into the field and looked at clinical samples that on the first 30 patients, we did get a couple misclassifications, but as we keep adding more patient samples to our neural net, now we can get very high classification accuracy for knowing which sort of drug a patient should be treated with. And then final project I'll mention is looking at tuberculosis and drug susceptibility of tuberculosis. This is a slightly more challenging problem, in part because TB has various antibiotics it could respond to, for example, rifampicin, isoniazid, moxiflacin, and amikacin. And usually it takes TB about 40 days to culture. So if you test positive for tuberculosis, it's very easy to know you have TB. But it, of course, takes a long time to know which type of TB you have. It takes about 40 days for a lab to determine which antibiotic you would respond to. And in the meantime, you're either suffering in the hospital or you don't want to be suffering. So a physician would normally give a patient basically broad spectrum antibiotics. And that, of course, is giving rise to antimicrobial resistance. So the question was, could we basically determine which drug the TB would respond to just by taking a spectra of a single TB that happened to be in a sputum sample and then identify which antibiotic it would respond to. So what we found is collecting Raman spectra, we could get kind of nice grouping here. Each point kind of corresponds to a different spectra. It's almost a projection of that spectral information onto a two-dimensional plane. And with over 87% accuracy, just by looking at the TB spectra from a single cell, we can identify which antibiotic it would respond to. So I didn't show some images here, but we've actually built now kind of a miniaturized microscope that We'll be part of a nonprofit that I'm founding with my students Loza and Baba, also in collaboration with Manu Prakash, basically to deploy this to the global south for more rapid TB diagnostics. So I will end there. That was kind of a quick foray into the world of nanophotonics and how you can control light at the nanoscale to be able to identify very rapid reads of biological specimens. And I kind of quickly showed you nucleic acid detection, metabolite detection, and then label-free bacterial detection. And I think there's a pretty exciting future in our ability to monitor both environmental and human health using kind of the digital technology that has transformed how we communicate with each other. Hopefully it can allow us to kind of better understand our own health and optimize that of the planet. So thanks so much, and I'm happy to take any questions.